Okay, good afternoon. This is Nelly Poyak from Dana Farber. Welcome to our seminar series that's hosted by the FHCC and Leukemia and Lymphoma Society. Today is April 20th, supposed to be a day of the Boston Marathon, but unfortunately that's all canceled. So just as a reminder, the talks are uploaded on the website, the FHCC-Harvard.edu website, so you can listen to the talks later on in case you have a conflict. And we had issues sometimes with using Firefox as a browser. So if you have a problem, then just try a different browser. And also as a reminder, we have Q&A at the end of the talk, and please use the Q&A function on Zoom to enter your questions, and we will try to answer as many as we can live, and then the speaker will have access to the questions, and she can get back to you later on in answering this. And with that, I'd like to welcome today's speaker, Shirley Liu from Dana Farber. Shirley is a professor of biostatistics and computational biology at the Department of Data Sciences at Dana Farber Cancer Institute and Harvard T. Chen School of Public Health. She's also professor of statistics at Harvard University. And today, Shirley will talk about a computational modeling of tumor immunity. Thank you, Shirley.
Great. Thank you, Nelly, for the introduction. And it is my honor to give this Connect Science seminar and share our work on computational modeling of tumor immunity. So here is my disclosure slides. Cancer immunotherapy has brought paradigm shifts to cancer treatment, and Science magazine ranked cancer immunotherapy as the top breakthrough of the year in 2013. In 2018, the pioneers of cancer immunotherapy development were awarded the Nobel Prize in Medicine. When we started cancer immunology explorations, there were little tumor profiles on immunotherapy trials available in the public. So we turned into these tumor profile cohorts. Even though none of these tumors were treated with immunotherapy, we still hope to learn something about tumor immunity. Among the many different profiling platforms, we focus on tumor gene expression because RNA-seq is both widely used and also getting cheaper and more cost-effective. First, we wondered whether the tumor bulk RNA-seq can be used to infer immune cell infiltration in the tumors. And we developed a computational method timer for this. Recently, we have updated the timer website, which we pre-computed the immune infiltration of all of the TCGA tumors. So users can explore the association of gene expression or mutations on the gene with tumor immune infiltration, and also how together these factors influence patient survival. The web server now also allows users to upload their own gene expression profiles and use 6 different computational algorithms to infer the tumor immune infiltration levels. And this is already published work, so I won't mention too much. You can explore this. Next, we want to see whether the tumor RNA-seq data contain immune repertoire information. Our immune system is able to recognize antigens, either external or tumor antigens, by generating very diverse immune receptor repertoires through VDJ recombination. For example, in T cells randomly select V, D, and J segments and join them together to create the diverse immune repertoire. The VDJ junction also have additional indels and mutations, which further increase the repertoire diversity. This hypervariable sequence on the T cell receptor is responsible for antigen recognition. Immune repertoire profiling is commercially available, but the assay is very expensive and consumes valuable material in the tumor biopsies. So we looked into tumor bulk RNA-seq. Since T cells and B cells in the tumors express TCR and BCR. Unfortunately, the hypervariable CDR3 sequences are not in the germline sequence. So most of the CDR3 sequences from the tumor RNA-seq are unmappable to the reference genome. However, since V and J have some sequence patterns, we developed a computational algorithm, TRUST, to specifically examine the unmappable reads from the tumor RNA-seq and de novo assemble the TCR and BCR repertoires. So here is our earlier work on the computational methods and analysis of TCGA, TCR, and BCR repertoires. We continue to make improvement to the TRUST algorithm. The current version is TRUST-4, which has improved, you can see from speed and memory efficiency improvement. And also we achieved a better precision and recall compared to another computational method, MyXCR. The algorithm can also infer more complete CDR3s. You can see in this, we have the complete CDR3, also the full-length VDJ sequence, and constant region assembly. And so with the TRUST-4 algorithm from TCGA, we are able to assemble about 70 million TCR and BCR CDR3 sequences, including about 34 million complete sequences from TCGA. Previously, there were computational methods for calling TCR and BCR from SmartSeq single-cell sequencing data. TRUST-4 can call TCR and BCR not only from SmartSeq, but also from 10x Genomics 5' single-cell RNA-seq. This is a comparison of TRUST-4 calls from just the 10x single-cell RNA-seq. This is using the 5' single-cell RNA-seq kit. And in comparison, we look at the TCR and BCR calls from 10x VDJ experiment. You can see basically the TRUST-4 result and the 10x VDJ results really match very well. This is another example where we have the 10x single-cell RNA-seq from PBMC, but there is no matching 10x VDJ experiment. You can see that TRUST-4 can make TCR and BCR calls from most of the T cells and B cells, which agree with the cell type annotation by Surratt. It can also call this small population of delta gamma T cells in here next to the CD8 T cells. Since the 10x genomic single-cell RNA-seq, the 5' kit has similar price, and the experimental procedure is also quite similar to the 3' kits that people traditionally use. We would recommend people to always use the 5' kit, because you do the experiment the same way, you can still get the single-cell gene expression, but with the 5' kit, you can get the TCR and BCR computationally for free without doing the 10x VDJ experiment. From the assembled TCGA BCR sequences, we observed 1.2 million BCR CDR3 clusters like this. Basically, from the same tumor, we might observe multiple CDR3 sequences. They share very similar sequence with just occasional mutations in here. This is because when BCR encounters antigen, it will activate the B cells for clonal expansion and create somatic hypermutations to evolve antibodies with better specificity against the antigen. So this is an indication of antigen recognition and B cell clonal expansion in the tumors. How do we know that the mutations we caught here are real somatic hypermutation instead of calling errors? We can look at the overall distribution of the somatic hypermutations we call. First of all, you can see that within all of TCGA, the assembled BCR CDR3 sequences are on average about 14 to 15 amino acid in length. And this is the somatic hypermutations we observed on the BCR DNA sequences. We can see this very strong 3 base pair periodicity on the synonymous codon. This is because the 3rd base on the codon is not under selection, but the first 2 nucleotides really can change the amino acid sequence. This suggests that the tumors really have selection pressure after somatic hypermutation during antibody evolution. In addition, tumors with higher AID expression also have higher BCR somatic mutation rate. So there is a question, how does TRUST-4 deal with situation where single-cell RNA-seq doesn't cover the whole length of TCR alpha-beta RNA? This is going to be a problem. We can, for single cell, we try our best to make the calls, especially since you don't have the accompanying 10x VDJ experiment for TCR or BCR. Some of the assembled CDR3 sequences will be partial CDR3s. So when B cells encounters antigen, in addition to somatic hypermutations, the BCR also switch its constant regions, also known as isotypes, in order to call help from other immune cells. An analogy of this is if you were to call 911, you'll get ambulance, firefighter, and policeman. Because TCGA have RNA-seq data mostly in the paired end sequencing format, we're able to find the different VDJ sequences with their corresponding constant region in majority of the TCGA tumors. And sometimes we can see multiple BCR sequences. They share either the same or very similar VDJ, but they have different isotypes. This again indicates Ig class switch recombination, which is another evidence of antigen recognition. So IgA is known to be produced by plasma cells in the mucosal tissues to insulate our immune cells from antigen in the GI tract and lung. And so we do observe higher IgA in gastric cancer, colon cancer, and rectal cancers. However, in many cancers that are not really of the mucosal tissue, we also observed high IgA B cells. If you look at the order of isotypes on the chromosome, we hypothesize that IgM is mostly naive B cells and also the early clonal expansion, and then it's shifted to IgG, and there is this watershed in here. We hypothesize that the isotypes before have more anti-tumor activities, whereas the isotypes after have less anti-tumor activities and potentially even pro-tumor activities. For example, in tumors of the non-mucosal tissues such as bladder cancer, ovarian cancer, and melanoma, we can see that IgG3 and IgG1 level divided, this is normalized by the total B cell level, is associated with better patient survival. So how is this achieved? One important B cell immunity mechanism is complement-dependent cytotoxicity, or CDC. Basically, when antibody recognizes the tumor antigen, it binds to C1q, which in turn recruits the membrane attack complex. And MAC will punch holes on the cancer cells for cell lysis. So in TCGA, we can use C1q expression as a proxy for CDC activity. And you can see it is positively associated with these isotypes before the watershed, but less associated or even negatively associated with these isotypes after the watershed, suggesting that indeed IgG1, IgG3 are probably more associated with CDC. Another B cell immunity is antibody-dependent cell-mediated cytotoxicity, or ADCC. Basically, when the antigen recognizes the tumor antibody, the antibody recognizes the tumor antigen, it interacts with the Fc gamma receptor, or CD16A, to recruit natural killer cells to the tumor microenvironment for cancer killing. In addition, the neonatal Fc receptor on antigen-presenting cells also help to stabilize the antibodies from proteasome degradation. So in TCGA, if we use CD16A expression as a proxy for NK activity or ADCC, you can also see that it is positively associated with IgM and IgG3, IgG1 before the watershed. But it's negatively associated with IgG4 and IgA. In addition, tumors with higher IgG1, 3, and also higher FcRNA expression not only have higher NK cell infiltration, but also higher NK cell activity using CD16A as a proxy for ADCC. So these data all indicate that B cells in the tumors indeed are recognizing tumor antigens. They have clonal expansions and they evolve better antibodies and also use CDC and ADCC to kill the cancer cells. And so you wonder, if that is the case, why does tumor still persist? And so to answer this question, we look at another system. So blood is another non-mucosal tissue, right? If we examine the healthy donor blood, this is again using trust to call TCR and BCR from the blood RNA-seq of healthy individuals. You can see young people, children have more naive B cells with IgM. As people age, their hematopoietic cells have lower ability to generate naive B cells. And prior antigen exposure also shifts the isotype from IgM gradually to IgG, then to IgA. Interestingly, in AML, this is a myeloid leukemia, we do see clonal expansion in both the T cells and B cells. In addition, we see that B cell class switch is much more and also earlier from IgM to IgA. You can see the infant already look like an adult healthy individual. And from children or adults, this shift happens even more. And in the adult, majority of the B cell antibodies are of the bad isotypes, right? So what is the consequence of this? You can see that in infants and children, if they have more IgA, because they are still mostly IgA1, then these infants would have worse clinical outcome. In adults, we see more shift to IgA1 and even IgA2. You can see when the adult patient has more IgA2, they have worse survival. Michael Karin's lab previously published studies showing that TGF-beta can induce IgA class switch recombination, which is a potent immune suppressor through PD-L1 secretion. So we examined these AML samples and we found that indeed TGF-beta is associated with the IgA levels in AML. However, this IgA level is not significantly associated with PD-L1. Instead, it is significantly associated with FOXP3 level, suggesting that the IgA plasma cells might recruit Tregs and are associated with a suppressive tumor microenvironment. So to summarize this part, we developed a computational tool, TRUST, which can infer the infiltrating T cell receptor and B cell receptor repertoires from tumor bulk and single cell RNA-seq. We also observed from TCGA tumors that B cell clonal expansion and IgG3, IgG1 are associated with B cell mediated immunity, especially CDC and ADCC in the tumors. We also observed IgA to be associated with TGF-beta, Treg, and worse clinical outcome. This suggests that initially in the tumor, B cells are recognizing the tumor antigen. It starts clonal expansion to evolve better antibodies to kill the cancer cells. But the tumors can secrete cytokines and force the B cells to shift into IgA antibodies, which will then have pro-tumor activity. Therefore, we hypothesize that if we give back the more antigen-specific IgG antibodies, because B cells can shift, antibodies cannot shift themselves, then they could kill the cancer cells. So there's a long question in here regarding the different proportion of antibody classes in non-tumor versus AML samples in adults and children. How do we know there is a class switch event happening and not just a preferential expansion of certain antibody classes? Is it important to do lineage tracing experiment to support the class switch hypothesis? Very good question. I agree, some of them we don't know the exact lineage. However, when we have a somatic hypermutation cluster like here, in here, if we see this from the same tumor, we see the clonal expansion, they share very similar sequences. And based on the IgG and IgA, we will know the order of the isotype class switch order, and then we can get the lineage. Especially in TCGA tumors, we have the germline sequence, so we know the order depending on the level of mutation from the germline. Okay, so next we want to also use the tumor RNA-seq data to help us predict patient response to immune checkpoint inhibitors. So here we stand on the shoulders of giants and get good insights from the review by Thomas Gajewski, where he proposed two broad categories of tumor immune evasion. In the first type, cytotoxic CD8 T cells are infiltrating the tumors. So these are usually called immune hot tumors, but the T cells are dysfunctional or exhausted. In the second type, CD8 T cells cannot infiltrate the tumors, so they are immune cold tumors. Ideally, we want to model these two types of immune evasion using large cohorts of tumors treated with checkpoint inhibitors to compare the difference between the responders and non-responders. However, since such data is very limited in size, we decided to look at public data from treatment-naive tumor cohorts. Even though these tumors haven't been treated with immunotherapy or any drugs, they still can give us insights on tumor immunity. For example, in the TCGA melanoma samples, we see cytotoxic CD8 T cells are in generally good for patient survival. So if the patient have higher CD8 T cell toxicity, then the patient usually have better survival. But this could be conditioned on the expression of other genes. For example, TGF-beta is a very well-known immune suppressive gene, and high expression of TGF-beta is known to cause T cell dysfunction. And indeed, we see that when in the melanoma tumors, when the TGF-beta expression is high, CD8 T cell has no benefits on patient survival. It's only when TGF-beta expression is low, we see the strong beneficial effect of CD8 T cells on survival. And of course, this is just one gene that we know already. We then developed a regression-based method to unbiasedly test every gene in the genome to see whether their expression can interact with the CD8 T cell levels in the tumor to influence patient survival. From many public tumor cohorts, so here we look at 33,000 tumor samples in over 180 tumor cohorts, we saw these 5 cohorts to give very robust signatures of this interaction. And so these genes are fairly consistent, you can see, in the different cohorts, and so we just average them to get a more robust signature. And indeed, these signatures agree well with hits from other T cell dysfunctional studies in the mouse. And so we want to emphasize that this T cell dysfunctional signature is a genome-wide signature. Then, given the expression data from a tumor, we could correlate this expression data with the genome-wide T cell dysfunctional signature to reach a single value, which indicates the level of T cell dysfunction in the tumor. This is not subject to multiple hypothesis testing and more robust against noise on the individual genes. Okay, so so I have a number of questions. TCGA data may like expression data on the stromal. Have you looked at this? Yeah, so the stromal part is more in the T cell exclusion side. Another question is, what about CD4 Th cell role in this? Yeah, so we do look at the CD4 T cell infiltration. But if you look at many of the cohorts, first of all, T helper cell estimation so far is not very robust, because out of all the CD4 T cells, T helper is only a small population. And in order for us to use a particular cohort, we first need to see, in this case, we first need to see that CD8 T cell cytotoxicity levels are associated with survival before we use this cohort to test every gene. And so far, we haven't seen consistent results where the CD4 level can, or at least the T helper level, can give us very consistent survival benefits. More specifically, do you find the, the immune repertoire question, maybe I'll ask at the end of the seminar, because we have passed that page. And so basically, to continue here, we have this genome-wide T cell dysfunctional signature. Once you have a tumor expression, we can correlate tumor expression with this genome signature to evaluate how dysfunctional the T cells are in the tumor. Next, we look at the stromal, which is related to T cell exclusion in the immune cold tumors. Here again, Thomas Gayaski's review gave us very useful insights. He listed a number of suppressive cell types which can prevent T cells from infiltrating the tumors. And so we extracted the expression profiles from Gene Expression Omnibus to generate signatures for those immune cell types. And among the different cell types we tested, 3 cell types really consistently give us phenotypes of T cell exclusion. So these are myeloid-derived suppressor cell, M2 macrophage, and cancer-associated fibroblast. You can see this is TCGA melanoma again, where each dot represents one tumor. The x-axis indicates whether this tumor has very strong expression correlation with each of the suppressive cell signature. And the y-axis indicates CD8 T cell cytotoxicity. And so you can see that when a tumor has high level of MDSC, M2 macrophage, or cancer-associated fibroblasts, they usually will have lower CD8 T cell activity. And so when we average the signature of these 3 together, we get an even more robust signature. And we have tested this against many solid tumor cohorts and found the signature to robustly predict low T cell infiltration in all of the cohorts. Again, this is a T cell exclusion signature, is a genome-wide signature. Given the expression data from a tumor, we can correlate this tumor expression with our genome-wide T cell exclusion signature to reach a single value indicating the level of suppressive cell types preventing T cell from infiltrating the tumors. So putting these 2 together, we developed a computational model called TIDE, or Tumor Immune Dysfunction and Exclusion. TIDE predicts patient response to checkpoint inhibitors from the pretreatment tumor expression data by evaluating both T cell dysfunction and T cell exclusion. So basically, we're asking, if this is an immune hot tumor, are the T cells too dysfunctional? But if this is an immune cold tumor, are there suppressive cell types preventing T cell from infiltrating? There are another questions. How about in other tumors? Are those same 3 cell types? So in most of the tumors, these 3 signatures are correlated. There are some exceptions. We are currently evaluating such data and also looking into the tumor single-cell RNA-seq data to see whether there are certain cancer types, only maybe 1 or 2 of those suppressive cell types are more indicative of tumor T cell exclusion. But overall, when we use the average of the 3, we can get a pretty robust T cell exclusion signature.
Dr. Liu, feel free to keep going through your talk if you like, and we'll see how many questions we can fit at the end. Thank you.
Great. Okay, thanks. So TIDE works like this. Given tumor expression, TIDE first decides whether this is an immune hot or immune cold tumor by evaluating the level of CD8 T cells and also cytotoxicity levels. And if this is a hot tumor, then TIDE will correlate the tumor expression with the genome-wide dysfunctional signature to evaluate whether the T cells are exhausted. Let's look at this example for anti-CTLA-4 trial on melanoma, which was published by Levi Garawai's group. You can see these groups, these tumors are the immune hot tumors, and if they have a high level of T cell dysfunction signatures, the patients are less likely to respond to anti-CTLA-4. But if in the original tumor we evaluated this, it turns out to be an immune cold tumor, then TIDE will correlate the tumor expression with the T cell exclusion signature to see whether there are suppressive cell types preventing T cell from getting in. And back to this cohort, you can see these are the CTL low or immune cold tumors, and you can see here when there are suppressive cell signatures in the tumor, patients are unlikely to respond to checkpoint inhibitors. In contrast, cold tumors without these suppressive cells could still be responders, maybe because anti-CTLA-4 could stimulate T cells for tumor infiltration. And this is also in agreement with recent published papers from Howard Chan's lab, suggesting that immune checkpoint inhibitors don't just revive the exhausted T cells within the tumor, but help circulating T cells to infiltrate the tumor. And so now if we look at the TCGA melanoma samples here again, each dot is one tumor. The red dots are the immune hot tumors, and the blue are the immune cold tumors. And the x-axis evaluates T cell exclusion, and the y-axis evaluates T cell dysfunction. Even though the TCGA melanoma patients were not treated with checkpoint immunotherapy, our model could still make some predictions, right? So these tumors, yes, they are hot tumors, but their T cells are more in a dysfunctional state, so they are less likely to respond to immunotherapy. These patients, not only they have immune cold tumors, but they also have suppressive cell types preventing T cells from infiltrating the tumor. And so these patients probably also don't respond to immunotherapy, and you might need to use additional drugs to help the turn tumor into an immune hot tumor before immunotherapy can work. Whereas these patients in the middle, they are likely to respond to checkpoint inhibitors, right? So this is our prediction. When TIDE was published, we evaluated on a number of the publicly available immune checkpoint inhibitor cohorts. In recent years, many different immunotherapy response biomarkers have been proposed, although many are evaluated or derived only on very limited cohorts. As more and more immunotherapy cohorts are being published, the data becoming available, we are now developing TIDE as a website to integrate these public tumor cohorts and also biomarkers together. And so for this TIDE website, we have collected the following published tumor cohorts. And even though all of the journals require the tumor profiles to be deposited and made publicly available, you can see we are not able to get all the tumor profiles. And these are the published biomarkers, and we were able to either get the code directly or implement some of the features available. And of course, we will continue to improve and also add new cohorts with time. And so with that, TIDE has 3 different features. The first function for TIDE is a regulator prioritization function. This is really for cancer biologists. For example, anytime you do a genomic experiment, such as a CRISPR screen or a single cell experiment, there might be hundreds or tens of hits. And cancer biologists usually can't know from just the limited data which of the genes are really important based on the availability of all the public data. And so we use TIDE to really help with this prioritization. And so this is an example where we use close to 700 druggable genes annotated by the OASIS database. And when we enter all of the gene here, it automatically gives us a rank order. And the top gene is PTPN2. This was recently published as the top hit in Nick Henning's paper, where he used in vivo CRISPR screen to identify this gene as an important regulator of tumor immunity. And so we can see here, these are the 5 T-cell dysfunctional cohorts in here. And the PTPN2 is not really inducing very strong T-cell dysfunction, but when it's high, you can see immunotherapy patients usually don't respond very well to immune checkpoint inhibitors. At least the red indicate that these are bad for patient survival. Interestingly, when these are all the immune-related CRISPR screens, we can see when PTPN2 is knocked out in general, the tumor are shrinking, is more sensitive to T-cell and NK cell attack, or in vivo tumor, it's shrinking more. And the last 3 columns are indications of whether the signature is high in cancer-associated fibroblast, MDSC, or tumor-associated M2 macrophage. But you can see if you have a genomic experiment with many hits, you can just enter onto TIDE and use the public data to help you prioritize. If you have only one gene, you can also enter this one gene. We can show you how this gene behaves in 180 different tumor cohorts that are non-immunotherapy trials as well. The second feature of the TIDE is the biomarker evaluation. This is mostly for translational scientists. For example, if some group just derived a new immune checkpoint inhibitor response biomarker from limited trials, they can then specify these are my biomarker. You can give a weight, or without a weight, we'll just average all the gene signatures together. Then we can see how well this new biomarker is performing in light of all the publicly available immune checkpoint inhibitor trials. You can also compare your own signature or biomarkers with other published immunotherapy response biomarkers. And the third feature of TIDE is response prediction. This is really for oncologists. So if you have a cohort of tumors, whether or not the response information is available, you can upload the tumor gene expression profiles, and we will use not only TIDE, but many other published immunotherapy biomarkers to make the prediction. We are not saying that TIDE have to be the best, right? So you can see that this is the TIDE prediction, this is the actual response. We are not perfect in prediction, but supposedly if a patient is predicted by many different biomarkers as a non-responder, then maybe he should be treated with a different drug. Whereas if every biomarker predict the patient to be a responder, then they have a high chance of responding to current immunotherapy. So we have some questions. What about the nature of the tumor itself, mutation burden of tumor in deciding the T-cell dysfunction and exclusion? Very good question. So in general, we focus more on gene expression. I think tumor mutation burden or new antigen load is indeed a good biomarker. It has been used in many tumors studies or checkpoint inhibitor trials, but we believe that this is only one aspect of tumor immunity. If you want to see the overall immune cell infiltration, whether the T cells are dysfunctional, whether there are other immune cells there, mutation is only one angle. We do think that expression provides very, very informative orthogonal data. And another question is whether TIDE can relate with protein function. Unfortunately, so far we only have gene expression. I think the protein data, usually people evaluate very limited number of proteins, such as PD-L1 level, or maybe you can stain for CD8. And these are not necessarily uniformly done in all the trials. So far, we did not use that. We just look at the gene expression. And also for these trials that have the TMB information, we did include that. Okay. Yeah, so I hope that the work I've shown you, you can see that tumor RNA-seq, even on the treatment-naive tumors such as TCGA, is cost-effective to inform tumor microenvironment and also tumor immunity, especially considering that the price for RNA-seq continue to decrease. You can now just isolate RNA, send it to a facility or a company. They can do library preparation and sequencing for like $200 to $300 samples, right? So it's actually a very, very informative data to tell you about tumor immunity. And in light of this, as part of the NCI Cancer Moonshot, we are in the process of developing a comprehensive tumor RNA-seq analysis pipeline. And we look forward to making this pipeline available to the community so that when you have tumor RNA-seq data, you can also use this pipeline to look at many aspects of tumor immunity. Of course, we don't just look at RNA-seq. So recently, as we mentioned, as part of the NCI Moonshot, we are in a CMAC-CIDC network within the NCI. And so NCI will coordinate the immuno-oncology trial centers to send their trial samples to 4 cancer immune monitoring and analysis centers, or CMACs, for comprehensive molecular profiling. So depending on the sample availability, we might profile tumors, for the first level of genomics, some imaging, proteomics, and then just depending on availability, we can do the higher or the more complicated assays. And so for this, Dana Farber has the Cancer Immune Data Common, which is the data center and will be responsible for creating the database infrastructure, computational pipelines, and bioinformatics algorithms to facilitate correlative analysis with the CMAC and IO trial centers. And these efforts are now being also supported by the foundation of NIH, where pharmaceutical companies also join forces to share resources and data. And our goal is to identify biomarkers for optimizing immunotherapy strategies in cancer. In addition to cancer immunotherapy biomarkers, we are also hoping to identify new regulators that will modulate the tumor immunity and enhance immunotherapy efficacy. In our original TIDE paper, we found this SERPINB9 gene as a regulator for T cell dysfunction. So SERPINB9 is normally expressed in T cells and NK cells in order to protect T cells and NK cells from granzyme B leakage. So SERPINB9 is a very potent granzyme B inhibitor. Interestingly, from the protein atlas data, we found that SERPINB9 is also highly expressed in the cytoplasm of cancer cells, which suggests that cancer cells have hijacked a T cell or NK cell mechanism in order to resist granzyme killing. And previously in our published CRISPR screens in the cancer cells that are co-cultured with T cells, we observed that when we knock out the SERPINB9 in the cancer cells, it can really improve the T cell mediated killing of the cancer cells. Also, in other published studies, we have observed that SERPINB9 is upregulated in the post-treatment of immunotherapy trial samples using single-cell RNA-seq. We know that this is upregulated in cancer cells. And also, this is a mouse tumor that is resistant to checkpoint inhibitors. The data above here is in human tumors. So we do see that SERPINB9 expression is one way for regulating the T cells, regulating the cancer cells against T cell attack. Unfortunately, SERPINB9, even though it's a regulator, is not a good target to improve immunotherapy response, because if we were to inhibit SERPINB9, the tumor immunity will kill both the cancer cells and T cells and NK cells, which is undesirable. So we look into other potential targets here. We know that in the tumors, altered glycosylation induce immune suppressive signaling. And so using this TIDE website regulator prioritization function, we input all the genes from the N-glycan biosynthesis pathway and let the TIDE prioritization module give us a top hit. And interestingly, you can see here, this MAN2A1 gene is the top hit. We then experimentally validated that indeed, when we knock out MAN2A1 in the cancer cells, they are more sensitive to T cell and NK cell mediated killing. So MAN2A1 is an enzyme that can increase complex glycosylation. So we try to knock out the MAN2A1 cell and then do proteomics profiling to look at glycosylation. And so you can see here, when we knock out the MAN2A1, it decreased the complex glycans in the cancer cell and also increased the simple and hybrid glycosylation patterns in the cancer cell. We also generated RNA-seq data in the cancer cell lines that are knocked out of MAN2A1. And you can see here, MAN2A1 is the most significant downregulated genes. We then use this downregulated gene to generate a signature and use this signature to evaluate across the immunotherapy cohorts to see whether this is associated with patient response to checkpoint inhibitors. And what we observed is that there is one cohort, this is the Mariathison cohort published by Genentech, and this is a tumor treated with anti-PD-L1. And we see that basically tumors with lower signal of MAN2A1 seem to have much better survival benefits. We did not observe MAN2A1 knockout or this signature to be associated with PD-1 or CTLA-4 response. And so to validate this, we knocked out MAN2A1 in the mouse tumors and then treat the mice with anti-PD-L1. And you can see basically after we knock out MAN2A1 in the tumor, we can see a significant increase in CD45 cells, CD3, CD8, and NK cell infiltration. In addition, looking at the tumor RNA-seq in the in vivo tumor with MAN2A1 knockout and anti-PD-L1, we can see in general increased antigen presentation pathway. This is antigen presentation and inflammation response and also T cell cytotoxic activities. And so in general, the tumor is more inflamed and having more tumor killing activities. And then we also look at whether this can shrink the tumor or make the mouse live longer. And for that, we tried both the MAN2A1 knockout and also a small molecule inhibitor, swansonine, which inhibits both MAN2A1 and MAN2A2. And you can see here, basically with the MAN2A1 knockout, even though PD-L1 itself or MAN2A1 knockout itself does not seem to have a very strong effect to tumor growth, this combination is quite potent to reduce tumor growth and also improve the mouse survival. Same is also observed with swansonine, this small molecule inhibitor. By the way, this drug has already been through IND process, but it failed a phase 2 clinical trial in tumors. But you can see here, this is in the tumor when we knock out the when we treat the tumors with either swansonine alone or anti-PD-L1 alone, it doesn't have effect, but this combination is very potent to decrease the tumor growth and also increase the mouse survival. And so in summary of the work on TIDE, we have created this website that you can try, and this TIDE algorithm can model T cell dysfunction and exclusion to predict patient response to checkpoint inhibitors. We also now utilize the public tumor cohorts and many biomarkers to help other biologists to prioritize genes, evaluate other new biomarkers, and also stratify patients. And using this TIDE gene prioritization function, we also identified a potent combination therapy between MANTO-A1 inhibition using Swanson-9 small molecule inhibitor and anti-PD-L1 treatment, suggesting that PD-L1 unglycosylation might be important for tumor immune evasion. So I have a number of questions in here. Do you confirm the importance of intratumor hypoxia and in particular adenosine metabolism as a negative predictor of response to immunotherapy? This is a very good question, Bill. We should definitely look into this. I don't think we just started looking at tumor immunity and hypoxia. We should definitely check this. The MANTO-A1 signature has many downregulated glycolytic genes. Any idea how MANTO-A1 is affecting glycolytic metabolism? I would say glycosylation, at least to a computational biology group like us, is very, very complicated. We are not saying that PD-L1 glycosylation is the only thing that's influenced. Many other things do get influenced, as you can see, increased antigen presentation, which might be responsible for increasing the immune cell infiltration and the T cell cytolytic activities. Another question, during the tumor growth, I believe that gene expression will change as well. Yes. How to group data from different time points? Well, biomarker evaluation response prediction be different? That's a very good question. Unfortunately, depending on the cancer types, you can see here, right now the TIDE website has less than 1,000 tumor samples that are in immunotherapy trials. Most of these samples are collected before immunotherapy treatment. After immunotherapy treatment, it's actually very, very difficult to get the patient samples unless it's like head and neck cancer or melanoma or new antigen trial samples, because if the tumor responded and you have a complete responder, you have no tumor to even do a biopsy. But if the patient did not respond, it progressed, unless we have additional treatment that we can give to the patient, ethically, there's no reason to get their tumor biopsy again to do tumor profiling. So the post-treatment data, yes, it's extremely valuable, but it's very limited. Are you planning to do a prospective study where you predict response based on their immune profile pre-treatment? Unfortunately, we are not starting our own trials, but that's why we are working with the Cancer Moonshot Project to use all the existing data to evaluate the prediction biomarker. I think with more and more trials from the CMAC profiling efforts, we will gradually refine the biomarkers to make better and better predictions. So did you test the TRUST and TIDE algorithms in mouse syngenic models? Yes, we have a study that we currently submitted looking at the CD26 tumors, CD26 syngenic model in the mouse tumor. Interestingly, with CD26, when you treat with checkpoint inhibitors, some mice, even we bought the mice from the same company, some mice do better and some mice don't. And what we found is that there are cancer cell intrinsic mechanisms, which means that the expression level of some CD26 clones are different from other clones. But there are also tumor extrinsic mechanisms that suggest that the mice, even though they are kind of age matched, lifestyle matched, they still probably have different innate immune health, that some mice just respond better than others. So which is really interesting, and we can see definitely T cell and B cell clonal expansion in the tumors in the responders, and also the TIDE prediction algorithm was very good at predicting the T cell dysfunction and exclusion. Other questions, does TIDE identify subcluster in each cell type? How robust is it in identifying specific subclusters of one cell type important to immunity? I'm not as clear about the subtype or subcluster. Look at another question. Have you checked the influence of MANTO-A1 inhibition on other T cell co-inhibitors like TIM3 and also which is also unglycosylated? That's a really good question. We Well, actually, we can definitely look at the tumor expression to see whether TIM3 is also influenced in terms of gene expression. I don't think, so in terms of glycosylation, the reason we suspect that PD-L1 glycosylation is important is that we only see this synergy for PD-L1 with MANTO-A1. We do not see this synergy for PD-1 with MANTO-A1, which makes us suspect that at least PD-L1 and glycosylation might be one aspect of this synergy. Okay, so yeah, so in the current world of personalized precision cancer medicine, I think personalized combination therapy is the future. Targeted therapy is still very, very important. It works by specifically targeting cancer mutations to kill the cancer cells. And it's still probably the main workforce for cancer treatment. Cancer immunotherapy is mostly to boost the patient's immune system and indirectly kill the cancer cells. We are always interested in cancer epigenetics. I'm at the Center for Functional Cancer Epigenetics with Miles Brown at the Dana Farber Cancer Institute. And we see that when any cancer cells are treated with drugs, whether targeted therapy, chemotherapy, or immunotherapy, we see a lot of epigenetic genes to be upregulated or at least mediate tumor response. So a lot of the epigenetic regulators seem to mediate the system balance in terms of stress or drug response. And so these three are very complementary in their mechanisms, so potentially can be combined for better tumor treatment. But in order to do this well, we need continued increase of tumor profiles. Even though with TCGA there are 10,000 tumors profiled, if you really want to look at any specific mechanism of, for example, HER2 patients with a specific other mutation, there are very few tumors still available to really give us a robust result. So we need continued tumor profiles, and CRISPR screens are also very, very effective because it gives us a functional readout to see when you really knock out or inhibit a gene, what is that effect. And of course, all of these generated high throughput data, and so we need good computational models to really integrate these data together in order to really have a cure for cancer. I know this moonshot goal is very, very ambitious, but we'll continue to work on this. So I want to thank many, many members in my lab. A lot of them have already graduated and started their own labs. It's with their hard work that we're able to develop the tools for the community and make these exciting discoveries. And I also want to thank many of my collaborators within the Dana Farber Cancer Institute and also in the rest of the Harvard community, and also other external collaborators. And I also want to thank funding agencies for funding support. And we are a computational biology lab, so besides the TIMOR, TRUST, and TIDE, we also develop many other computational tools to help people model gene expression, transcription, epigenetic regulation, do CRISPR screens, targeted drug response, and hopefully these tools will also be very helpful for others. So I want to thank you for your attention. I see another question. Are we able to recognize the range of cytokines such as IL-2, IL-33 in colon cancer by these computational tools? Which tool is highly accurate in analyzing them? If the cytokines are directly expressed in the tumor, whether it's colon cancer or other cancer types, I think the tumor RNA expression does give you at least some reasonable proxy for their expression. However, IL-6 in the blood seems to be also an important biomarker for immunity in general. And that's a protein circulation level. I'm not sure whether the blood RNA-seq will actually give us that indication.
Well, Shirley, that was just fantastic. Thank you so much. And you've left absolutely nothing for us to do at the end in terms of tossing questions to you. It's right at the 1 o'clock hour, so your timing was also perfect. I want to thank everyone for joining us. As always, please feel to send us any additional questions or comments. Tomorrow, we're very fortunate to have Dr. John Dick from Toronto, speaking on heterogeneity in leukemias and effects of treatment. And we'll continue from there through the rest of the week with Dr. Tim Rebick on Thursday, talking about prevention and early detection. So thank you all again for joining, and everyone, please be well.
Great. Thank you.